in last class we had discussed about how we proceed for diagnosis, right? So the first step was the pre-analytical stage, in which the patient approaches a clinician. The clinician then, based on the signs and symptoms, then thinks some of the provisional diagnosis with the differential diagnosis, and then sends some of the sample for either confirmation of diagnosis or exclusion of diagnosis. And in that regard, we had learned something about what is known as what is the sample, what would be, how the sample should be collected for analysis. And we ended that session with how we should send the sample in a proper way. Now, thereafter, today we will start discussing the next step. So, the next step would be the analytical part or the analysis. Now, what kind of analysis do we do for the diagnosis? The first step of diagnosis would be, we look for the pathogen directly. Okay. If we look at the causative agents, we have very specific causative, either bacteria or a fungus or a parasite or a virus. So, in those specimens, we look directly. How can we look directly? We can make a mount of the specimen and observe it under the microscope. That would be the first step. It is also called as direct mount or wet mount. Sometimes we need to add some other agents so that we can identify them more in a better way or more clearly. For example, there is a simple mounting called iodine mount. What happens in that? We use a solution of iodine to the sample. Now, why this is important? Eventually, we will learn when we will be discussing about parasites. Some of the parasites have glycogen molecules in the cells. So, glycogen molecules will react with iodine. That is a basic chemical reaction you have all done in school, and it will observe, we can see it more clearly. That is a very simple. Similarly, we can use some of the staining methods. Staining means we colorize them. So, when we do staining, we can appreciate even better. Have you understood this? If something does not have a color, we can observe it, but when we do impart some coloring agent to it, the picture becomes much more vivid. Now, when we talk about staining, the staining can be either positive staining or it could be negative staining. Just if you have seen the negative prints which were used to be available earlier in photographic films, that is, when we look at the photograph that has been developed, it looks beautiful with the color, with the person vividly in color. But if we look at the negative print, what will happen? The background will be in distinctly color, but the person in focus would not be. So, in positive staining, we try to stain the organism itself, and if we do it other way around, that is, the background is stained but the organism is not stained, then we call it as negative staining. Okay. So, there are many methods. Again, we can use something called as impregnation method. We can use some molecules which will be specifically taken up by the organism and then stained, or we can use a particular method in which some particular type of organism will be stained in a particular color, others will be stained in a different color. So, what it helps is, we can differentiate between two types of organisms. It is called as differential stain. That means, with this staining method, we can differentiate between predominantly large group of organisms. The other method would be what is known as antigen detection. Now, what is antigen? Antigen is a protein. Okay. Antigen means protein which can stimulate an immune response. Now, if it is a microbial protein, then it will stimulate an immune response. By that way, we can also detect, right? For example, let us consider a bacteria A. It has entered into the human body. What will happen? This bacteria A will have some proteins. These proteins will stimulate production of antibody. Now, in the early phase, what will happen? The body will have lot of proteins of this bacteria. Then the body will slowly start producing antibody, and the body will get rid of the bacteria. Now, based on this phase, we can detect the presence of organism either by looking for the antigen or the antibody which specifically acts against the antigen. In the early phase, when the body has not yet reacted to produce antibody, we can find the antigen in huge number. But when the body starts producing antibody, we can look for the antibody. A very good example will be like a dengue fever, that is very common. Right in the early phase, we look for something what is called as NS1 antigen. That is a non-structural protein. So when in the early phases, the person will have viremia, that is, the virus is present in the blood. If we test for the antigen of the virus, it will come positive. Slowly the body will start producing antibody response and the virus will be cleared. Now if I go and look for antigen, we will get a negative result. Now we have to look for antibody. So this is the basic concept of serological diagnosis. Okay. Furthermore, I think you have already read in immunology, the immune response will then slowly get matured. Initially it will be of IgM type, then as the immune response matures, it will be in the IgG type. So if we detect IgM, we can say this is probably an acute infection or infection that has happened very recently. Whereas if the response is of IgG type, we can confidently say the infection is not recent. It may be remote or a long term. That is the method of antigen or antibody detection. Another method, I think you have read in biochemistry, we can detect for the nucleic acids itself, be a DNA or an RNA. So we can directly look for the presence or absence of nucleic acids of the pathogen in the sample itself. Have you understood this? I think it's not a very there are some particular tests. These are called as nucleic acid amplification test with the use of a method called PCR or polymerase chain reaction. We look for particular segments of either DNA or RNA depending on what type of nucleic acid the pathogen has. For example, a virus may have either DNA or RNA, a bacteria will have a DNA. We look for specific sets of bacterial DNA. Now, if this DNA is present, that means the bacteria is present. Okay. This is also even very helpful for diagnosis, especially in certain scenarios like conventionally we talk about bacteria will grow in culture. But in some of the situations, the bacteria does not grow in culture or the bacteria is present but it is dead. In those case scenarios, we can detect the presence of the organism with the help of detection of the nucleic acid. Whether the bacteria is dead or alive, it doesn't matter. If the nucleic acid present, then we can detect. Now, all these are various methods, but still today, the crux of the diagnosis is culture, identification of the organism, and getting the susceptibility test done. Why? Because in all other methods, we can diagnose the case. Then what about management? We need to manage the patient also. What antimicrobial agents has to be given? Only this can tell us. For example, for the same disease, let us take two tests. Enteric fever is a disease caused by salmonella. During the phase of bacteremia, bacteremia means during the phase when the organism is present in the blood. If we take the blood for culture, we can get the organism in culture, right? Few days later, when the body starts producing antibody, we can get a serological diagnosis. Now consider the two facts. In the second phase, we have done a serological diagnosis. We can certify that this is a case of enteric fever. But can we dictate the management in this case? No. We can only give an empirical treatment. What is empirical treatment? The treatment that is suggested based on the community response. But they may or may not work for a particular individual. But if we do get a culture, we can take the bacteria out, we can test the bacteria for the common antimicrobial agents that are used for treatment, and we can test them in the laboratory. Then we can say, okay, antimicrobial agents number 1, 2, 3 will act and 4 and 5 will not. Then when you are the prescribing physician, you will say, okay, then I have to choose between these 1, 2, 3 antibodies. This is the importance of culture identification and susceptibility testing. Now, when we say culture, that means we are trying to culture in an artificial medium in which we can test. Now, what is this media? It is also called as a culture media. It could be a solid or liquid substance which will allow the growth of microbial agents. There might be various microbial agents based on their requirement. There are many types of culture media. Now, what does this culture media have? They will have what are the needs of organisms? Now, it's a very big question. Let me simplify it. We human beings are only a small fragment of life form on the face of the earth. Microorganisms are present in huge number. Among all of the microorganisms, a very small fraction of microorganisms actually comes in contact with human body, and among them even a smaller fraction actually cause disease because they have evolved with us, and all those which cause human disease will have some basic common things. That means they are dependent on nutrition and they are dependent on temperature. So most of them will grow at body temperature, that is around about 37 degrees centigrade. Okay, and among the nutrition, what will be the nutrients? Obviously it will need water, electrolytes, protein and carbohydrate source. Simple. So if we do need to make a culture media, that is an artificial substance on which this bacteria will grow, what do we need to put it there? We need to put water, we need to put electrolytes, we need to put proteins in it, we need to put carbohydrates in it, and we need to put it at 37 degrees centigrade, right? Now, to make it solid or liquid, we need to put another things, what is known as agar agar. What is agar agar? It's a seaweed which has a long chain polysaccharide. Now, this long chain polysaccharide at room temperature becomes solid, but when it is heated to about 50-55 degrees centigrade, it melts to become a liquid. Now, what will be the advantage of having a solid culture? We can take a look at the colony, the bacteria which grows, we can isolate them and we can segregate them. If it is in a, say in a broth, that is in a tube, there are multiple bacteria in it. We cannot identify them or separate them. On a solid culture, we can do that. Okay. It may have many sources of protein like a meat extract or an yeast extract or even blood or serum. For example, I think you have heard about a culture media called as blood agar. Why it is called blood agar? Because it has got blood. Similarly, there might be an yeast agar which contains yeast extract. It could be a RCM, that is Robertson's cooked meat media, which will have meat particle. There are various types of media. I am not going into each and every specific, but you need to read some of them, a common. Now, coming to the type, it could be either solid, semi-solid or liquid or broth. Then it could be either a conventional, that is the manual of the how things have developed over the years, or it could be a specific culture media used for automated systems. Now, what are automated systems? Automated systems are obviously computerized systems which will do the work of a human being. It is much less laborious. Okay, so some of the common media, I will just discuss them, you can read later on. First is what is known as simple media or basal media. Simple media means it simply will have water, electrolytes and some basic protein. It will help to sustain the organism. Organism may or may not grow, but it will help in sustenance. The simple basal media will have peptone water, nutrient broth or nutrient agar, or could be a semi-solid media. These are examples. Now, problem with them, some of the bacteria will have more specific requirement. If it has specific requirement, it will not grow. Similarly, we have for them what is called as enriched media. Now, when we say enriched means we are adding some extra things. It could be blood, it could be egg, it could be yeast extract. That means we are trying to enrich the media for some of the bacteria which will normally not grow on simple media. So, blood agar is a very good example of enriched media. Next would be if we take the blood agar and we heat it at an appropriate temperature, what will happen? All the protein within the blood will be semi-digested. So, some of the bacteria we cannot digest the proteins in the blood, they will able to grow, they will be able to grow. So, this is even more better for fastidious organisms. This is called as chocolate agar. Another very good example would be Loeffler's serum slope, in which we use serum for enrich. Now, if we take a broth and put some of the protein extract in them, we get what is known as enrichment broth. So, enrichment broth is for isolation of specific organisms. Why? Let us say we are looking for a particular bacteria causing diarrhea. Now, the stool sample will have millions of bacteria. If we directly put it on a culture media, say enriched or a basal media, what will happen? Almost all will grow. But the pathogenic one, we may miss it. So, we need some media which will allow the growth of a particular specific bacteria. That is called as enrichment broth. Okay. For example, a tetracycline broth for Salmonella typhi. If we take it the other way around, that means we add some chemicals which will inhibit the common organisms, but will allow the pathogenic one to grow. That means we are inhibiting other organisms. Then we call it as selective media. So we can make it selective by very commonly with the use of antimicrobial agents. Some of the examples will be Lowenstein-Jensen media. This media is used for culture of tuberculosis bacilli. Now, tuberculosis grows very slowly and we use sputum for culture. Now, when the patient gives the sputum, some of the bacteria of the oral cavity will come. So, we need something in the media which will inhibit the oral bacteria but will not inhibit tuberculosis bacilli. Some of the antibiotics are used over there which makes it specific for Mycobacterium tuberculosis. So, this will be an example of selective media. Now, coming to the last type of media, it is called transport media. In the last class, I have already told you what is the purpose of transport media. The purpose of transport media is to what? It will preserve the organism when we are transporting the clinical sample from the ward or hospital to the laboratory. Okay. It may not promote the growth, but it will preserve the organism. It will not let it die. That is the objective of a transport media. There are many types of transport media for specific organisms. Okay. When we will go and read about those specific organisms, we will discuss the transport media. This is important about culture. Now, after having done culture, we need to differentiate. So, one of the media is differential media, like differential stain. So, what is the purpose of a differential stain? It differentiates between two sets of organisms. Similarly, differential media will allow us to differentiate between two sets of organisms. Let me explain. An example of differential media would be McConkey agar. Now, this agar contains only lactose as carbohydrate. Some bacteria will be able to utilize the lactose and some would not be able to utilize the lactose. And it will also have an indicator. What happens is that the bacteria which is able to utilize lactose will form lactic acid. The lactic acid will change the pH and the indicator will give color. But if a bacteria cannot utilize lactose, what will happen? There will be no acidic environment, there will be no change in color. Thus, we can easily differentiate which bacteria is a lactose fermenting and which is lactose non-fermenting. thereby helping us to differentiate between lactose fermenters and non-lactose fermenters. Similarly, based on other metabolic elements, we may have used clay agar or other as differential media. Another group of media is very important, that is known as the anaerobic media. Till now, whatever we have talked about, all the bacteria that we are culturing in aerobic environment. However, a set of bacteria are anaerobic. That means they grow in absence of oxygen. That means we cannot keep it in an incubator openly. So we need a specific separate setup. One of the media is a RCM, other would include like glycolate broth, and sometimes we may also use, okay, I'll come. There are many anaerobic system like Gaspec system. I think it would be better if we talk about this thing when we are talking about anaerobic bacteria. Otherwise, it will become too much information. We have separate sets of media in a separate incubatory conditions, all of which will maintain one thing, that is, it will make the condition anaerobic in nature. Okay, now coming to blood culture. Yesterday I had discussed, last class I had discussed about the way we take the blood for culture. Now we can do it in two ways, either conventional way or in an automated way. The conventional way in which we can use a blood culture bottle which may have media either monophasic or biphasic. Now look at this picture. In this bottle there is a broth, whereas in this bottle there is a broth along with an agar slant. We can use either of them. The advantage of the biphasic media is that if we don't have a biphasic media, every time we try to look for whether the organism has grown or not, we need to do a subculture. And every time we try to do a subculture, there is a risk of contamination. But in biphasic media, what is the advantage? We don't need to open the bottle. Every time we need to do a subculture, we can just slant the bottle, the media will go over the solid agar, and we can observe if there is growth or not. Hence, it is much easier. Even then, conventional blood culture is laborious and it takes a lot of time. Hence, in today's time, most of the laboratories have moved on to automated blood culture systems. Now, what are automated blood culture systems? They are basically machines in which we use these sort of bottles that is made by the manufacturer, and this bottle will have a disc at the bottom of the bottle. Now, importance of this disc is that this will have a pigment. When the concentration of oxygen in the solution decreases, this disc will change in color. So what the machine does is, it detects the change of color of the disc. Now if a bacteria grows, oxygen concentration will decrease, the disc will change in color, and the machine will say, okay, this is the bottle which could possibly have bacterial growth. So what will be the advantage of automated blood culture system? We don't need to do repeated subculture, and the machine can detect the change a lot earlier than what a human being can. There are many types of automated blood culture system like bacterial or bacterial. Okay. Now coming to culture methods, commonly we use the blood and McConkey agar in most of the sample. However, we can use a clay agar or a high chrome agar for urine or any others when we are suspecting something. Sometimes we may look for fastidious organism which may cause meningitis. For that which we have to use a chocolate agar. So as and when required, we need to change the type of the media which are used. Now how we inoculate the media? Very commonly we use a hood. This is called a biosafety hood. Last class I had already discussed about the biosafety levels 1, 2 and 3. Based on that, we use a biosafety level, commonly biosafety level 2A, and in that hood we use metallic or disposable plastic loops to inoculate the culture media with the specimen. And when we do inoculate, after overnight we can observe growth of the bacteria like this on a culture plate. And if it is in a broth, we can observe turbidity in the broth, sometimes surface pellicles or in precipitates. If the bacterial colony has a higher density, it will precipitate. If it has a lower density, it will form pellicle. Thereafter, when we get the bacteria in the colony, we identify it and then we put up what is known as susceptibility testing, in which we can do a lawn culture, that is, we get an uniform growth, not a single colony. Other methods for biochemical analysis would be pore culture, stroke, and stab culture, and we incubate in either an incubator or a BOD incubator, commonly at 37 degree with a error range of 2 degrees centigrade. And in case of anaerobic, we can use a candle jar and other methods of anaerobic, which would include gas pack system. This is an anaerobic glove box. Now, coming to identification. Based on the colony, how the colony looks, what are the pigments, what are the size, and on blood agar, we can observe something very important, which is known as hemolysis. Some of the bacteria, especially the pathogenic bacteria, will have some enzymes which can digest blood. Based on that, we can divide whether it's a alpha hemolysis, beta hemolysis, or gamma hemolysis. What is alpha hemolysis? This is a partial digestion. That is the reason why you can see a kind of a dirty greenish hue against the colonies. Whereas in beta hemolysis, you can see clearing against the colonies or complete hemolysis. And what is gamma hemolysis? That is no hemolysis. So alpha would be partial hemolysis, beta would be complete hemolysis, and gamma would be no hemolysis. So most of the pyogenic bacteria, pyogenic means which causes pus, they will have some enzymes which are hemolytic. Thereafter, we need to identify what type of bacteria is. How we identify it? By studying its metabolism. What do I mean by metabolism? What are the enzymes they produce? What are the carbohydrates they can utilize? What is the main mechanism of their oxygen utilization? What is, whether it's fermentation or oxidation? All these things will tell us whether this particular bacteria belongs to which group. We can do it manually, and again, in today's age, we have automated systems. So, some of the example is, for at least at your level, for gram-negative bacilli, we have some test called indole test, citrate test, urease test, sugar utilization test. For gram-positive, we have catalase test, oxidase test. These are some of the tests. I am not going into the details of each and every test. When we will be studying about some the important bacteria, we will learn about this test more. So, the basic outline is that we study the metabolism of this bacteria and try to identify them. Phytic system or Phoenix is another system in which the same principle is utilized, but the identification is done by the machine. Hence, the turnover is quicker, identification is faster. One more method of identification is known as MALDI-TOF, that is matrix assisted laser desorption ionization time of flight. I am not going into the details of the methodology. It basically analyzes the type of protein present in the bacteria. Different bacteria will have different spectrum of protein. Based on that spectrum, it can identify. Advantage is it identifies organisms more correctly, more quickly. Disadvantage is the machine is very costly. After that, now we have cultured the bacteria, we have identified it. Now we need to know which of the antimicrobial agents will be effective against this bacteria. That is, we need to do antimicrobial susceptibility testing. Now, this type of testing again can be done conventionally and it can be done by automated methods. So, conventionally what is done is that There are two ways. One is known as disc diffusion. Disc diffusion means on this culture plate, we place some of the discs containing the antibiotics and we place on the plate. What will happen? The antibiotic will diffuse from the disc into the media. And we have the organism plated on the culture plate. If the organism is susceptible, what will happen? We can see a clear zone of inhibition around the disc. However, if the organism is not susceptible, it will grow further into it. So, based on the zone, we can identify whether the organism will be susceptible or resistant. Now, keep in mind, whatever we are doing it in vitro. That means in the laboratory. It will give us a predictable outcome, but it will not dictate the exact outcome. What I am trying to say is, suppose you have some sort of an infection. I have done the culture, I have identified the bacteria, I have done the susceptibility testing. I can say in all probability a particular antibiotic will be effective, but that doesn't mean it will always be effective, because the antibiotic we are giving in vivo, that means inside the human body, and we are testing over here in vitro, that is in the laboratory conditions. The other method would be to use what is known as broth. Over here we are using a solid agar, solid culture media. If we are using broth, what will happen? As the concentration of the antibiotic agent will increase, the bacteria will die and we can see clearing of the broth. This also gives us a concept called as MIC, or minimum inhibitory concentration. I will talk about MIC, MBC later on when you have a further understanding of the disease. Just keep in mind, broth dilution gives us MIC. This will be very important when you learn a little bit more. When we will start talking about how to use antibiotics. Okay. Now, as the picture suggests, doing the broth dilution will be more laborious. Huge number of tubes for each and every antibiotic. The alternative is an automated machine which can do that for it. So it will be less laborious, faster result outcome, and this is also known as E-test or epsilon test. What this test is done? We take a solid culture plate, plate the organism over it, and then we use this kind of strip, also known as E-strip. Over here, the antibiotics is present in a graded manner, that is according to concentration. We look at the inhibition. The point where the inhibition starts is the MIC. So this is a easier way to get the MIC. I will talk about MIC and MBC later on. Okay. So what will be the outcome? For a particular antibiotic, this bacteria will be either susceptible or will be resistant, or it could be intermediate or susceptible, dose dependent. As far as today's class is concerned, keep in mind it could be either sensitive or it could be resistant. When we'll talk about antimicrobial susceptibility later on, I'll discuss about intermediate and susceptibility. Next, I have already told you, with the use of serological tests like precipitation, agglutination and ELISA, we can detect either antigen or antibody. Based on the duration of the disease, we can identify. Among the molecular methods which either detects the DNA or RNA, we may have used PCR, that is polymerase chain reaction, and the variants of PCR, which may include a real-time PCR or a reverse transcriptase PCR. That means reverse transcriptase, what it does, it converts RNA into DNA and then we detect the DNA, especially in case of viruses. Or we can use what is known as biofire film array. What is biofire film array? This is a nucleic acid test. Normally in all the nucleic acids, we are looking for one organism at a one point of time. When we are talking about film array, we are testing for multiple organisms with the molecular test. Okay. So this is even better, especially in some of the clinical samples like CSF, where the load of the organism will be very low even with the clinical presentation. Or it could be a cartridge based nucleic acid test. What is a cartridge based nucleic acid? That means the whole test procedure has been reduced in such a form that there are specific cartridges in which the whole test is performed. That means technical expertise is not required to perform this test. So there are many variants of nucleic acid amplification test. And finally, the last part of identification would be microbial typing. What do you understand by typing? Any idea? So there are many types of typing. First type would be serotyping. What is serotyping? A particular bacteria will cause immune response. Now both the antigens will be different based on the different antigens of the same bacteria. If it is differentiated, we call it as serotype. It may have different ribozymes. It will be ribotyping. These bacteria might be affected by different types of phage viruses. It is called as phage typing. So there are many types of typing. Why we are interested in this type of typing? A very important application would be for investigation of an outbreak. Okay, try to listen to this one. Suppose there is an outbreak of a particular infection caused by a, say, Klebsiella, a gram-negative bacteria. Now Klebsiella is present almost everywhere. Almost every human being will have some sort of Klebsiella. But when the outbreak occurs, only a particular strain of Klebsiella will cause the outbreak. So if I get 3 patients who are all infected with Klebsiella, how will I know whether it is the same strain or not? Then we need to type it. And if after typing it comes as the same strain, then we can say, okay, this outbreak is caused by this particular strain of Klebsiella. Okay, so this is called as typing. So that would end the overall overview of laboratory diagnosis in infectious disease. I hope you have got some idea how we proceed. Okay, so for final summarization, it would be pre-analytical stage in which we examine the patient, look for the clinical diagnosis. Based on the provisional diagnosis, we go for analysis. What would be the purpose of the analysis? Either to confirm our provisional diagnosis or to exclude other diagnoses. Then we need to, in the post-analytical phase, we need to implement the results of the test. If it's a confirmatory test, we need to manage the patient based on that test. Okay, and if it is an exclusionary test, we need to follow up the patient or do other tests based on that. Now, in our department, among the analytical tests, how we can identify? First would be direct identification. In direct identification, we can look for how? We can look for the organism itself under the microscope with or without staining, using various types of staining. It could be positive staining, negative staining, differential staining. We could look for the presence of antibodies or antigens. Antigens means microbial antigen. That is, we can look for serological diagnosis. Third part would be we need to extract the organism itself. Why this is very important? Because if we can culture the organism itself, we can test them against antimicrobial agents. That will help in dictating the management or the treatment of the patient. If we cannot do that, the alternative method would be to look for the nucleic acids that are present. This is especially helpful in organisms like viruses, which are very difficult to grow, some of the bacteria which do not grow. In all these case scenarios, the nucleic acid amplification test helps. There are many types of nucleic acid amplification test. And finally, there is something called as typing. The typing helps in determining whether a particular strain of the organism is causing an outbreak or an epidemic, etc. etc. I think you have already heard about during the COVID pandemic, there were some particular strains. Even in the news, they were circulating that this strain is now circulating. How do they do that? It is by typing. There are many methods of typing. It could be from serotyping to DNA typing to phage typing in virus. There are many types. Okay. So that concludes the overview of laboratory diagnosis. Have you understood? Henceforth, we'll look at some of the important aspects of this diagnostic process individually. Okay.